lot. It's my greatest pleasure to be here. Oh, this is working now. That's great. So thank you, Brian. Thanks to the rest of the organizing committee for having me here. I think it's great to be in touch with the community of aging researchers in Asia, now that I'm in Asia. I'm still in Germany as well, so I will have my group in Germany until mid-2027, and I already started to set up a lab in Beijing, which is an absolutely great place to be. So these are just my disclaimers. I am the co-founder of a longevity company together with Vera Gorbunova. And we are trying to use knowledge from these wonderful creatures such as naked mole rat, whales, and salamanders, which are the models that I work with, in order to identify longevity strategies. So I am myself a basic scientist. I increasingly got into translational research, and hopefully in China will be running my first human project. But until now, my lab has been leveraging salamander models in order to understand principles of regeneration and aging. And why these models? Well, because salamanders such as axolotls and newts are exceptional for their regenerative abilities. So these are the closest vertebrates to human, able to regenerate the widest range of complex structures that is known in nature. And this includes, for example, large chunks of their brains, large parts of their hearts, the tail, including the spinal cord, and they are the only tetrapod able to regenerate their near full limbs as adults. And these are some of the contributions that our lab has made towards understanding some of these processes. So besides this incredible ability to regenerate, what is outstanding is that these animals are able to invoke cellular plastic transitions in order to generate progenitors for regeneration. So they are usually generating progenitors by a de- and transdifferentiation, so not only by mobilizing stem cells. So in that sense, it's very different from humans, right? And the other thing that is notable is that this ability to regenerate persists through lifespan. And here I'm illustrating this with one of the axolotls in our colony in Dresden right now. She is 14 years old and is regenerating its limb. It's already reached something that we call the digit stage. So it's reforming its digit, and then this limb will just grow up in order to reach the size equivalent to the contralateral limb. Now, these animals have a lifespan of 10 to 13 years old, so this will be the equivalent of a centenarian axolotl, and yet is regrowing a full limb. So this takes me to another property that animals have, which is very remarkable, and this is negligible senescence. Oops. What have I done? There we go. So negligible senescence in nature is defined as the defiance of the Gompertz law of mortality, which was earlier described. So all studies done with salamanders of different species have led to the conclusion that there is not any changes in mortality through age. And not only that, but these are lifespan outliers. There is a remarkable lack of physiological decay with age. They have cancer-resistant, persistent fertility, and especially for the females. And this is a paper that we are now having given the final touches. And before you ask me in questions, no one knows yet what the causes of mortality for these animals are. So we started to use these animals to understand a little bit more of how aging happens in this context of enhanced regeneration. And remember, these are vertebrates. They have a very similar body plan to us, so it's very different from Hydra or Planaria, which are regenerative species, but are invertebrate. They are much more simple, and they regenerate by very different mechanisms. So in the case of the axolotl, we started to do a big exploration of different hallmarks of aging and how they manifest in these animals through lifespan. And the first one that we looked at is cellular senescence, which is a known hallmark of aging, and is a process in which our lab had made several contributions towards particularly understanding how senescence contributes to regenerative processes. And here, obviously, you know in this audience that senescent cells accumulate through time in various organs as mammals age. However, we've seen that this does not happen in salamanders. Now, you can ask what happens then with replicative senescence. And for this, we turn into looking at telomere biology, which was nicely introduced earlier during this section. And again, telomere erosion accompanies several age-related phenomenon in humans, and is accompanied as well by a silencing of telomerase activity in various tissues as they differentiate. However, we're not seeing this again in salamanders. So we covered the entire lifespan of these animals. I'm showing you here what happens with skin when it ages. But if you take liver, tail, mesenchymal tail tissues or blood, you will see the same thing. There's no signs of telomere attrition. And there's a basal level of telomerase expression in several tissues, for example, solid tissues, even at extreme ages. Now, we can look at this at the single cell level when we explore a single cell atlas of the axolotl after sexual reproduction. And here, what we would see is that telomerase is expressed in cell types that you wouldn't normally see necessarily in mammals, for example, in muscle and in neurons. And it could well be related to this ability to regenerate, as indeed we do find that axolotls, they need to upregulate telomerase every time they are regenerating structure. So we have seen it at least for limb and for tail as well. So this flexible telomerase activity, which seems to be keep going active through lifespan in several cell types, could be related to this ability to regenerate. Now, I will spend now a few minutes telling you about some important findings we had in the space of immune senescence, and in particular in the thymus, which is the first organ which starts to age in mammals. So this is obviously central for adaptive immunity, is where our T cells develop and they get selected. They learn what is self, what is non-self. And importantly, in all organisms known, the thymus starts to involute as we hit puberty. And this is a major component of immune senescence, and is not something that we can easily counteract, or we can counteract at all. So this is how the axolotl thymus looks like. It is 3 nodules at each side of the gill. So from the endodermal pharyngeal pouches, the axolotl bulges 3 nodules. Mouse or humans, they bulge 1, and they come together. In the case of mouse and human, it will be sitting next to the heart. So what is remarkable is that if we look at thymic nodules in axolotls from 3 weeks of age all the way up to 10 years, we will see that there's no signs of involution. So this is showing you representative sections at scale. There's no signs of involution and not signs of adipogenesis, as is obvious here. Now, what we did is to explore at the, actually I will go back because that's an important point. I would like to clarify that in the axolotl, sexual reproduction occurs between 10 and 12 months of age. So this is the point at which they should, in principle, if they were like us, start to involute. But they don't. So we look at what happens at the molecular level, and for this we built bulk RNA-seq of about 30 axolotl thymus covering the full lifespan. And what it became clear is that while there's a segregation in this PCA analysis of the young thymus, there is no segregation whatsoever with the adult thymic nodules. So there's a stabilization after maturation. And what we can do with this data set now is ask, the things that happen to our thymus as we get older, are they happening in the axolotl? For example, we can look at FOXN1, which is a master regulator of thymus epithelial cell identity, which is the niche for the thymus. And in a mouse between 1 and 3 months of age, would already decrease by half its expression. Now, what happens in axolotls, if we look at FOXN1, is an incredible stability, and only we see a little bit of decline in animals which are at the edge of the lifespan, 10-year-old animals. And what's more noticeable is that genes that are known to decline with age in the mammalian thymus, they are all known thymus regulators, things required for homeostasis or for training T cells, they are not coming down, they are still there. So this is quite remarkable. Now, as I mentioned, the thymus is formed by these important cells, the thymic epithelial cells, and when we age, they undergo a conversion to adipocytes. So we ask, is this happening in the axolotl? And the answer for this is no. So there is an upregulation of some mediators that we see here, but neither at the molecular, not at the morphological level, we're seeing any signs of adipogenesis. So recently, we also built the thymic atlas for axolotl lifespan, and this again enables us to test important questions, and we demonstrated that the composition of the thymus, even at the age of the lifespan, is not changing, and this thymus is still able to generate proper T cells at different stages, so it's able to perform continuous thymopoiesis. And the same, we have seen that proportions of the thymic epithelial cells are not changing. So these actually tell us, I'm going to skip this, that there's an incredible resilience in this animal for its thymus. It's the first example of a thymus that is not involuting. And what we are doing now is performing cross-species comparisons between species in which a thymus involutes versus the axolotl, and trying to test the candidates through collaborations in both mouse, zebrafish, and killifish. Now, the other question that you may have is whether this is due to an extreme regenerative potential in these animals. And actually, what we have recently found in my lab is that the axolotls are able to regenerate their thymus completely from scratch. So we are capable of de novo regeneration, which is the first example of whole organ regrowth in vertebrates, and it will come out in December very, very soon in Science Immunology. So this last question of whether these animals are not involuting their thymus because perhaps they have an extreme capacity to regenerate it, it is very, very important that it is central to this question of my lab, which is whether regeneration is akin to rejuvenation or not. And for this, we have been developing tools in order to assess biological time and as well as manifestations of aging. And one of these, as many of you have done, is to turn to the building of biological clocks. And for this, we paired with Steve Horvath, and we ask a simple question, which is, can we actually build an epigenetic clock for the axolotl? And in this context, we knew that it was possible to use the Horvath array, at least the conserved version of the array, in order to build clocks for xenopus. And xenopus are like the cousins of salamanders, are amphibians like the axolotl. But funnily, we are not able to build an axolotl epigenetic clock with these similar approaches if we take the entire lifespan of the axolotl. What we can do somehow is build clocks only up to 4 years of age, which covers this period which we call the early life of the axolotl maturation. If we try to build clocks from 4.5 onwards, nothing can be detected. So this is quite remarkable. And if we turn first to this first part, the part in which we can actually build clocks, what we see, I'm going to take a bit longer because I'm not going to do 15, sorry. We can see is that we can build dual clocks for xenopus and axolotls and for axolotls and humans. And indeed, we see that many changes that occur in methyl cytosines with age are going in a similar way between human, axolotl, and xenopus. So this tells us that somehow in this part of the clock, when they are aging, there are some similarities which are conserved between frogs, axolotls, and humans. However, we're still thinking what is going on with the rest of the clock. Why can't we build a clock? And for this, what we've done is to take a parallel approach to the Horvath arrays. And here we did unbiased methyl cytosine calling using nanopore technology of all promoters per sample. We're using promoters here because the axolotl genome is massive, is 10 times larger than the human genome, so we have to do basically adaptive sampling. But what was striking here is that we were not able to see signs of hypomethylation at the global level, nor signs of increased methylation at known sites, for example, at the polycomb sites, whose methylation levels increase in all, at least all studied species, including Xenopus, but we are not seeing that in axolotls. And what's more, when we focus on clocks, on promoters related to the genes that were observed were used in the array study, again we see a very striking stabilization of methylation after maturation in these animals. And we see this at the level of experimental gains in methylation or not. What we see is that there are no significant changes in methylation status, and definitely there is not a global hypomethylation in salamanders. So basically we think that axolotls deviate from the conventional notion of epigenetic aging. So they're quite remarkable in terms of this landscape. And not only this, if you say these animals have a stable methylome, therefore biological processes that relate to methylation status, you would expect them also to remain stable. And we see exactly that. For example, line 1 transcription, which is normally suppressed by DNA methylation and goes wary during aging, is relatively stable in many organs in the axolotl. And we see a similar thing in a collaboration here with Wei Dang from Baylor, that again, another process that depends on DNA methylation, which is the maintenance of a close chromatin and therefore cryptic transcription site closeness. We basically, this is a process that, again, gets dysregulated with age and results in cryptic transcription. And again, this is not happening in the axolotl as it ages. So, in the last actual data slide that I want to show you, I'll tell you that actually we have been able to use the clocks that work, so these clocks between 0 and 4 years of age in these animals, in order to ask this important question that I asked before, which is, what is the connection between regeneration and rejuvenation, at least at the epigenetic level? And here, what we've done is 2 experiments. The first one, we induce regeneration in these animals for 3 cycles, and then we compare the original limb in the same animals, which has aged, and the one that has regenerated 3 times. And what you can hopefully appreciate is that there is an epigenetic decrease in this age. This is DNA methylation age in limbs that have regenerated 3 times, compared with the contralateral controls that have never regenerated. So this would suggest that there's some level of rejuvenation associated with regeneration of the limb. What is interesting is that if you look at what happens in the tail, we are not seeing that. The tail seems to be aging together with the animals. And we think it is possible that this difference in rejuvenations versus non-rejuvenation events has something to do with the mode of regeneration. So we know from the work of many others in the field, as well as our own lab, that limb regeneration has major contributions from dedifferentiation of cells, whereas tail regeneration is largely achieved through stem cell-based mechanisms. So in the same organism, you have very different ways of regenerating. And we are now trying to look, using transgenic lines, at how is the level of potency in a context of natural regeneration related to the shifts in DNA methylation age and many other parameters that could be related to rejuvenation. So with that, I would like to just finish by saying that we have conducted a big multi-ohm study of axolotl aging, and we're now basically collaborating with a lot of scientists within the aging field. Our major collaborators are Stefan Rulans and Sid Horvath in order to deal with clocks and multi-ohms, but also we have a lot of other very, very interesting collaborations with different people in the field, including Peter, that obviously worked a lot with Brian. So with that, I'm going to thank my lab in Germany, and particularly these people that I highlighted in blue who are involved in the aging projects, and they are great. Our growing lab in China, our collaborators, funders, and I would like to also say that we are recruiting, so if anyone is interested, come and ask me, please. So we're recruiting at junior level, so in the lab, postdocs and bioinformaticians, but also we're recruiting at faculty level. So there's a lot of opportunities at CIMER, and if you do great research and high impact research, this will be a good fit for you. So thanks a lot.
Thanks, Max, for the nice. We have time for one question.
Two small versions of one question, I guess. I should know the first one, but what does the survival curve look like? What is the kinetics of mortality? They'd only live 13 years, and it's not an exponential increase, but what is it?
No, it's a complete drop all of the sudden. At least what we've seen in colonies.
It's like organismal senescence almost.
It's very, very difficult, very, very difficult to define. So most amphibians have a very big sort of like drop in survival at the beginning of their life, and then they remain super stable. So the problem is like no one has been able to study mortality of salamanders in the wild, and in the colonies, we had our colony, and I work with salamanders for more than 15 years, and I've seen 5 events of death. And the problem is that usually researchers don't have the money to support this type of studies. So now every time we see a natural salamander dying, which doesn't seem to have, by pathology research, doesn't seem to have anything else, we are collecting the tissues and seeing. But there's other organisms that do this, like termites. All of a sudden, after 20 years, the kings and queens, they dysregulate peewee, they dysregulate proteome, and collapse.
In the blood, there was a change in the methylation at the repeated the line elements. It seemed like, you said most organs don't change.
Yeah.
Is there something different about different tissues? Can you get to mechanism that way?
We are studying that right now. We're doing that right now, but overall, I would say it is quite striking the fact that this is not changing, and especially given that line elements, they comprise a big proportion of the genome as well. So largely the genome expansion is through line element expansions. Yeah.
Okay, thanks, Max.